Thanks. So it's great to see you all here and that we're all alive. 4 years ago, last week, the World Health Organization declared a pandemic. And now we're kind of back to normal, although there's still quite a few deaths each week from COVID. But it's a real great pleasure to introduce, I call him AJ, but Arinjay Banerjee. This guy's a very accomplished young ECR investigator. As you can tell, he's from India, and he's from Mumbai. And he's not from Mumbai, but he went to Mumbai for his master's at the National Institute of Virology, where they've got, is that the only or the one of the main BS4? BS4 facility, yeah. So then he came, he's got a Canadian connection from quite a ways back. He then came to University of Saskatchewan at Vido and did his master's there with Vikram, who's also a legend in the virology field, who's now retired and is in Victoria. And then, dear to my heart, he went to Australia for 5 months to Sydney, to the University of Sydney, where he did his One Health certificate training. So One Health is the integration of landscape and man and animals and infections, and how the changing landscape and the changing interface with frontiers and animals leads to the crossover. And so considering the whole planet and holistically, virology in that context, or infections in the context of the whole landscape and indigenous people, and not just as an isolate. So then he was working really passionately on coronaviruses and MERS. And MERS, as you know, this was the coronavirus 2 that came out from, that was infecting camels mainly in Saudi Arabia. But had that virus been the one that made the big jump to humans, that was a 30% mortality. And we'd be in a very different state now. I mean, that would have been one of those CEPs, which I've been told is a civilization-ending pandemic. I mean, 30% of the population was dying. So we were lucky. So then we're also lucky because then he went and started his postdoc with Karen Mossman at McMaster, and that she's also one of the legends. So he was there for 2 years when, quite fortuitously, the pandemic struck. Well, fortuitously for him. So firstly, I want to just cast back a little bit. But in his PhD, he got 11 first author papers, right? So this guy works hard. In his postdoc, he got 11 first author papers, but that was in 2.5 years. But in total, he got 40 papers in his postdoc. So also, it's part of being in the right place at the right time. He was at Mac, and then when the pandemic was declared, he went up to Toronto, and so he was living in Sam's Mubaraka's basement. And she was the one who was collecting samples from YVR, and she's also a fantastic virologist. He was living in the basement there. He set up the BL3 facility at U of T, and then they had 3 samples from YVR of patients who came in who were suspect. And 1 week later, this man here was the one who isolated the virus that many of you have been working with. And so the virus is designated the one that we all have been distributed to in Canada, SARS-CoV-2/S for Samara, and B for Banerjee for Sam, for him. But also that was like Sunnybrook. So this guy's quite famous. We've got a lot to be thankful for him and his fast work in helping the pandemic preparedness and work that would happen since then. So without further ado, well, actually, the 1 week set up for a BL3 facility contrasts what happened here at UBC, and we're still struggling with our BL3 facility. And that's how I met AJ, was through Karen, because we were working on like all of us, were all working on, became instant overnight virologists, working on the virus, and we needed to get into infection models. And so AJ did that for us. So it's a great pleasure to know you and to have you here, and we're looking forward to your talk.
Well, Chris, can you hear me? Yeah, I think Chris exaggerates quite a lot. It was stressful, and I'll tell you what, I'll start by saying that, no, maybe I'll use this. How about now?
Is that mic working?
I could switch to, oh, maybe I'll hold it. Yeah, this better? Okay, so our field of science been a lot of struggle for the longest time. No? Can you hear me at the back? Yeah. How about this? Yeah, okay, okay. This doesn't work, so I'll just use this. All the way up so Chris can hear it. Okay. How about now? Good? Okay. Good? Okay, good. So our field's been a lot of struggle. And when COVID-19 showed up, I think there was more appreciation for high containment science. And we're hoping to keep that agenda. So we continue to invest in science that looks at these pathogens. But what I'm going to tell you today is a story of how we struggled with developing some of the first models in the world to study coronaviruses in bats, and how that some of that work is now helping my own trainees study coronaviruses in bats. So I'll start by acknowledging the trainees. I have the privilege of standing and sharing all their work with you, but really they're in the lab working 24/7, well, not 24/7, but 18/7. And they're working very hard to get all the data that's helping us move the field forward. Very grateful to some of the funding agencies as well that continue to believe in the science that we do. So my lab is based at VIDO, not VIDO, but people think it's VIDO, but Vaccine and Infectious Disease Organization. So we're physically located on the University of Saskatchewan campus. And we've got Canada's largest high containment lab, 180,000 square feet. So that's an American football field inside BSL-3. To just give you examples of some animals we work with in BSL-3, bisons. So there's a huge Bison Parks Canada contract to develop vaccines against bovine TB. Of course, we love our bats. We do work with lots of bats, and we can house all sorts of small animal models. In theory, we can house any exotic animal species in BSL-3 to study a new zoonotic pathogen. We also have alpacas. That's our MERS coronavirus model because it infects camelids. We don't have camels in Canada, not yet. Of course, bisons and then bats. We've got white-tailed, well, regular deer. And then we've got pigs as well. We can have up to 1,000 pigs in containment for large-scale preclinical trials. During COVID-19, our vision was to emerge as Canada's Centre for Pandemic Research. We are expanding 25% of our BSL-3 capacity to house BSL-4 pathogens. And we have the space ready. We're just waiting for certification so we can go hot. We're building a new animal facility where hopefully I'll have 2 new bat colonies and non-human primates as well, and mosquitoes and tick colonies to study vector-borne infections. And we're also renovating a lot of other spaces that my colleagues are interested in. So within my laboratory, we call ourselves the Laboratory of Zoonotic Viruses and Comparative Immunology. We are 2.5 years old, very new. And we basically study largely within 3 themes. And today, I only have time to share stuff we're doing in theme 1. So looking at how highly pathogenic viruses interact with their reservoir hosts. We're also very interested, because we're a comparative immunology lab, we're interested in looking at how these viruses would also infect humans and what sort of different mechanisms we'd see in terms of immune tolerance and antiviral responses between the reservoir species and humans. And because we are based at VIDO, our mandate is to also help develop vaccines and therapies. So we are involved in some vaccine and therapeutic development as well, although that's not my primary focus, not at this point anyway. So before I start off with telling you why bats are cool, I'll start by telling you how diverse bats are. So some of you, or perhaps most of you, work with mouse models. It's really one species, Mus musculus, but you have different strains. Bats are over 1,470 different species. When I was doing my PhD, we knew of 200 species, And we've since added 270 species, and we continue to discover new bat species. So it's very hot right now. And if you break them down, we can break them into Gympterochiropterans, or previously they were called Megachiropterans, the big bats. So if you spread your arms out, that's how big their wingspans can get. And I hope you appreciate this young lady holding a big bat, a Malaysian flying fox, which is amongst the largest bats on the planet. And if you look at the small bats, the bumblebee bats, they would fit at the tip of your thumb. So the reason I'm showing you this holds true for our data as well. We don't make pan-bat statements. Depending on the species you study and the virus you study, it's very, very finely refined and tuned to that bat species. Some observations you could extrapolate, but we try and not make pan-bat statements. Bats aren't special. Some bats have specific adaptations. Bats can also travel long distances, so they do migrate. So bats in Canada, like our snowbirds, they go down south when it gets cold in Canada. In the summers, when we have lots of insects, these bats would fly back up north. So if a bat was carrying a potential pathogen, the bats also have the ability to carry that with them as they migrate. Bats are also extremely long-lived. Now, for mammals, our lifespans are directly proportional to our body size. So a larger mammal is expected to have a longer lifespan. So to give you an example, this bat, little brown bats, or Myotis davidii bats, they live up to beyond 4 decades. So 43 years old is the longest lifespan that's been documented for bats. Now that's fascinating because it's a tiny mammal. This bat is the size of a mouse, but it lives beyond 40 years. So that's something that's now triggered a lot of research into this field, trying to identify why bats have this exceptional lifespan. And we aren't doing this, but we've got excellent colleagues who are looking at telomeres and telomerases in bats, cell death, cell senescence. It's really cool. But this is why I got interested in bats and viruses in 2013. I was at the Max Planck Institute in Berlin as a visiting student, and my former PhD supervisor was giving a seminar at the Max Planck on bats and Ebola virus. Because 2013, 2014 was the big Western African Ebola virus outbreak. So I was sitting there as a just freshly undergraduate completed student, and I see this slide where Vikram talks about how bats can coexist with filoviruses like Ebola and Marburg virus, coronaviruses, severe acute respiratory syndrome, SARS-1, Middle East respiratory syndrome, porcine epidemic diarrhea, swine acute diarrhea syndrome, and now COVID-19. Paramyxoviruses that cause severe encephalitis like Nipah and Hendra virus, and of course, lysovirus, a classical example of which is rabies. Now, if you look at bats that are naturally infected or experimentally infected with these viruses, they don't develop signs of disease. They don't have symptoms of infection. So that really intrigued me when I was looking for a master's project that, hey, I want to study why bats don't get so sick with virus infections. And all of these viruses cause severe disease in humans and agricultural animals. So I walked up to Vikram and I was like, hey, Vikram, could I come to your lab and study bat innate immunity? And Vikram's response was very simple. He said, yes, but how would you do it? Like, I'll take some bat cells and I'll infect them with viruses. And Vikram at that point pointed out that there are no bat cells. So you start with making bat cells. And so that's where I started, and I'll get to it in a bit. But why are we so interested in coronaviruses? So if you look at all the coronaviruses that currently can infect humans, the top 4 coronaviruses are what we call seasonal coronaviruses. If you're immunocompetent, you'll get the sniffles for a few days and you'll walk it off, right? So these viruses we think evolved in bats or rodents. My cursor is dancing. There you go. Bats are rodents, and through intermediate hosts that we think we know and some that we don't currently know, the viruses made it into humans. If you look at the highly pathogenic coronaviruses, so SARS-CoV, MERS-CoV, and SARS-CoV-2, the viruses have ancestors in bats from which they likely evolved. Then through some intermediate hosts that we are confident about, like civet cats for SARS-1, camelids for MERS coronavirus, the virus eventually jumped into humans. For SARS-CoV-2, this is still debatable, but pangolins were considered an intermediate host 2 years ago, not so much today. But the idea is bats do carry coronaviruses that have the tendency to jump into intermediate hosts and eventually jump into humans. And we at least know of 7 examples of coronaviruses that have zoonotically transmitted themselves to humans. If you look at the pathology of infection with these viruses, we see a trend. There's dysregulated inflammation. Whether you look at porcine epidemic diarrhea in pigs, or you look at SARS, MERS, or COVID-19, it's really the dysregulated immune response that causes immunopathology. So with all of this in my mind, I had a very clear hypothesis from a master's project. I wanted to test the hypothesis that bats have evolved a process to control inflammation. If these viruses are driving an inflammation in humans, perhaps bats don't see it. So I started off, as Vikram pointed out, no tools. So in 2013, I started off by setting up bat cell lines. So we took Aptesticus fuscus bats. These are native to Canada. And we took lots of organs out. We tried setting up primary cell lines, but the kidneys took hold, so we ran with it. I set up 3 different clonal populations. And once we set up these cells, we wanted to see if viruses would actually infect them, because you can't do virology with cells that aren't susceptible or permissive. So at the time, I worked with Dr. Vincent Munster at the Rocky Mountain Labs. He had MERS coronavirus. We didn't have MERS coronavirus in Canada yet in an academic lab. So he tested our bat cells for infection, and I hope you can appreciate that we do see cell death and infection, along with the control cells, which were Vero cells from MERS, monkey-derived cells. We looked at VSV, vesicular stomatitis virus, from the same family as rabies virus, also propagates in all 3 bat cell lines, and our control human lung cell line as well. We also tested porcine epidemic diarrhea coronavirus, which is also a risk group 3 pathogen in Canada, and it grew in 2 out of the 3 bat cell lines. Why it didn't grow in the second cell line is of interest to us. We just don't have the time to go back and revisit this project, maybe one day. It does also infect the monkey cells as controls as well. And that's a video. As a master's student, I found it fascinating to make cell lines. So I made a video of the cell line, and we also landed up commercializing it. So if you wanted, you can buy it through CareFast in the United States. So what we've gone on to do over the last 10 years, and I'll summarize it with this slide because I want to show you data from my students' work. Over the last 10 years, we and our colleagues have clearly shown that in humans, coronaviruses drive a dysregulated immune response. We see a low antiviral response that's largely driven by the proteins in these viruses that can block type 1 antiviral interferon responses. We study interferon, so we'll continue to stick to that one protein. And we see an exaggerated inflammatory response in humans. What we've discovered is in bats, we see quite the opposite. The story is flipped. We now see a strong antiviral response, again, driven by the type 1 interferon response. I'll break it down for you in a bit what that means, and a low inflammatory response. Right? So bats somehow have refined the immune response against this highly pathogenic bat-derived coronaviruses. So at the time when I was just about wrapping up my PhD, we came up with a question on, can coronaviruses persist in bats? This was important because our disease ecologist colleagues in the field have identified coronaviruses in the bat gut. So our field currently believes that for bats, a coronavirus is a fecal-oral transmitted gut pathogen. It's not a respiratory pathogen, right? So we wanted to test this hypothesis using the cells we had developed. Can we get these cells to be persistently infected? So we took our bat cells, and at this point, I was being co-supervised by Daryl and Vikram. Daryl had just moved from the NIH lab to Canada to set up his lab, and I wanted to take my bat cells and mash it with his coronavirus expertise in 2017. So we infected the bat cells with a very low MOI, or a low dose of Middle East Respiratory Syndrome coronavirus, MERS-CoV. And I hope you can appreciate that over time, vast majority of our bat cells went to a massive cell death. But at about day 12, we started seeing colonies of resistant cells that started expanding in our cell line, on our Petri dishes. And over time, when we passage these cells, you started getting a monolayer of cells in your plates. So they survive, they grow, no cell death. But if you take the supernatant of these cells, the media that the cells are growing on, you start seeing varying levels of infectious MERS coronavirus coming out of these cells. And this stabilizes over time. After passage 2, we start getting this consistent amount of virus in the supernatant. But we still weren't convinced. We had this theory that perhaps some cells are infected that produce virions, and there are lots of resistant colonies of cells that aren't susceptible or permissive. So we wanted to go back and test that theory. So we took the cells and we stained them for the nucleocapsid protein from MERS-CoV. The mock-infected cells clearly don't have the green protein, which is the coronavirus nucleocapsid protein. We freshly infected cells as positive controls, and I hope you can appreciate that over time, we start seeing a bunch of green cells indicating infection. What we found in our persistently infected cells was rather very unique. We saw very small populations of cells that lit up as green, where we could visually detect the nucleocapsid protein. And very few cells in culture had lots of N protein, suggesting that these cells perhaps have more virus replication. So we concluded that looking at the protein was just not good enough. We had to look at viral RNA as well. So we developed in situ hybridization probes for MERS coronavirus nucleocapsid gene, and we stained every cell again. So the mock infected cells, they don't stain, so great, the probes are working. We freshly infected bat cells and they stain heavily red because we hit them with a lot of virus. We took our persistently infected cells and we now start seeing about 3 populations of cells, but every cell on our plate was infected. We saw some with lots of viral RNA, some with intermediate, and some with low levels of RNA. And I wanted to follow this up with testing my second hypothesis, what drives the virus persistence and shedding. But I had to move from my postdoc because my supervisor kicked me out. He said, it's good for you to move labs, go somewhere else, do something else. So we finished the study off and I moved on to McMaster for a postdoc. However, the first thing I did after coming back and starting my lab was put MERS coronavirus in vivo. So we took the same bat species. My colleague Paul Four at McMaster has a bat colony, a breeding bat colony of big brown bats. So I reached out to Paul and we'd become great friends while I was a postdoc, and he sent his bats over. And what Daryl and I did was we took about 9 bats and we hit them with MERS coronavirus. We don't really know a good route of infection for bats because we don't know how bats see coronavirus. We hit them with all 3 routes. We went intranasal, we went oral, and we went intraperitoneal, IP. Then to the 5th TCID50, we overdosed them with MERS-CoV. And over 34 days, they only gained weight. There was no weight loss, so weight loss would be suggestive of pathology, infection. And what we saw was very fascinating, and I'm hoping we're going to put this out very soon, and this is going to create a lot of debate in our field. We see infection in the lung for bats. That's never been reported before, because we believe that it's a gut pathogen. Because people haven't done this experimentally. You only go out, you catch a bat, and you look for a coronavirus. In an experimental setting, with our tremendous amount of viral dose, we see infection in the lung. But over time, this lung infection resolves. So by day 34, we start seeing very little viral RNA in the lung. What we do start seeing, however, is at day 34 post-infection, the viral RNA sticks around in the gut and some other organs that we looked at. So that is consistent with what our colleagues in the field observe, coronavirus RNA in the gut. But perhaps they're sampling bats that are 34 days out exposure period. And we don't know how long this viral RNA sticks around. So what we believe and what we're going to try to sell is we've now got a model of coronavirus persistence, which we can now start probing to study what causes loss of persistence and viral shedding in bats, subsequent human exposure, and potential outbreaks. But we've just got an AUP approved to extend this to 6 months. So we're going to extend this further down and see how long we can keep the bats infected. The only problem is the longer you keep animals in CL3, the more expensive your experiments become. So our limitation is really funding. But we're going to try and come up with a model for persistent infection bats. But when I started my postdoc, so I moved on to McMaster in Dr. Karen Mossman's lab. She spent 20 years studying a protein called RF3 in humans, interferon regulatory factor 3. And I'll get to the pathway in a bit. But for now, RF3 is absolutely critical for an antiviral interferon response. If you knock it out, mice die faster with virus infection. And this protein has 3 domains in it: DNA binding domain, RF association domain, because the protein moves as a dimer once activated, and a serine-rich domain. So at the time, I recruited a brilliant undergraduate student who I believe is now at UBC, Nicole, and she was co-supervised by Dr. Brian Golding as well and Dr. Don Bowdish. And Brian's a phenomenal bioinformatician. And what she did was she looked at every IRF in mammals, and she ran this positive selection analysis to see what's positively selected for in bats. So it's bats against a background of every other mammal. So in the serine-rich domain, RF3's got a bunch of serine residues, hence the name, serine-rich domain. When a virus infects a cell, these serine residues get phosphorylated, and that leads to activation of RF3 and subsequent downstream antiviral responses. So Nicole came back to me and said, hey, Arinjay, I found this additional serine residue at the 185th site of RF3 that's positively selected for in some bats. Is that important? And Karen and I were like, yeah, serine is very important for RF3 because it gets phosphorylated. So we went on and we developed CRISPR knockout bat cells. We were the first to deploy CRISPR knockouts in bats. And we knocked out RF3 in a bat background. We knocked out RF3 in human cells. And we then tested what Nicole had come up independently. She was unbiased because she didn't know what IRF3 meant. And we tested if this serine residue was indeed important. Again, we'll summarize this very quickly in one slide. So we discovered that the bat IRF3 with this additional serine residue induced a more potent antiviral response that shut down virus replication. The human IRF3, so we have IRF3, it is functional, it's not a bad protein, but we have a leucine residue at this site. And the response we see is we do have an antiviral response, but it's very modest compared to the bat version of IRF3. And finally, we did this classic experiment where we took the serine residue from bats, we stuck it into human IRF3, and we were able to demonstrate that significantly enhances interferon-dependent antiviral response. So this was the first time, and of course, we worked with my colleagues, Drs. Linfa Wang and Aaron Irving. They gave us additional bat cell lines to demonstrate that our observation was not cell type or species dependent. It was true for more than one bat species. And this was the first time really when we started thinking about how we could potentially translate these adaptations that bats have naturally evolved over 80 million years of evolution. Humans have evolved 200,000 years ago. We're a very new species on this planet. But bats evolved so many years ago, and perhaps we think they've seen these viruses for much longer than humans have. So having said that, I'll now break down this pathway for you. So what are interferons? What do interferons do? And how are we making discoveries in this pathway? So Victoria Gonzalez, our first PhD student in the lab, she started in 2021, and she took on this task of understanding how interferons signal in bats. So we and others have shown over the last 10 years that when viruses such as coronavirus infect bat cells, we do have sensors that can detect infection. They can detect viral RNA. They activate kinases in the cells, and then they turn on transcription factors, which then turn on interferon expression. So this has been done. We've shown this from multiple bat species. What the field doesn't know yet, and hopefully Victoria's paper is going to change that, is how do these interferons, once produced, signal and protect bat cells, and is that any different from humans? So we know from human and rodent studies that interferons, once produced, they interact with their cognate receptors, type 1, type 2 interferon receptors, so IFNAR1 and 2, and they activate a bunch of kinases, a set of another transcription factors, STAT1 and STAT2. When these transcription factors are activated, they move into the nucleus and produce interferon-stimulated genes, or ISGs. Now, interferons themselves are not antiviral, but the ISGs have tremendous antiviral capacity. And alone or in combination with other ISGs, they shut down virus replication. So Victoria asked, what would this pathway look like in bats? So the first thing we had to do again was, like my former supervisor pointing me in the direction of no bat cells, I pointed her no bat interferons. So you got to start by making the cytokines if you had to study those interferons. And she did this phenomenally well. So we used a Drosophila-based expression system where we were able to express human interferon beta. So we are studying type 1 interferons. We started with interferon beta, which is a type of a type 1 interferon. She made humans. She made Eptesicus fuscus, the insectivorous bat. And she also made Pteropus electo, the fruit bats found in Australia that are natural reservoirs for Hendra. So we've got two very divergent species, and we've got humans. We've got a vehicle control where she's just producing GFP as vehicle treatment. And what she did was then we took cells from these bats, the species matched bats. So the fruit bats got the fruit bat interferon, insectivorous bats got the insectivorous bat interferons, and the two human lines, the two human cell lines got the human interferons. And then we infect them with vesicular stomatitis virus that we've engineered to express GFP. So anything that's green is infected. Stuff that's less green is protected by interferon treatment. So when we leave the cells untreated, we see varying levels of infection. When you treat them with the vehicle, which is this GFP supernatant, we see no protection, which was expected. But then we start treating them with 10 units of species-matched recombinant interferon. And I hope you can appreciate that by 6 hours, we almost see complete protection in every cell type. So the interferon that Victoria is making in the lab for bats is functionally active, and it protects cells actively against a replication-competent virus. She's then gone on to do a series of different assays to demonstrate that transcription factors indeed move into the nucleus of these bat cells. So STAT1, STAT2 are functionally active, which is indicated by the red. She's also shown that STAT1 and STAT2 are also phosphorylated in bats. So we see clear phosphorylation of STAT1. We also see phosphorylation of STAT2. I don't have it on this plot. But what she's gone on to show is the kinetics are perhaps different. In bat cells, we see an interferon-dependent response much faster than we see in human cells. And that's the angle that she's currently chasing. And we also did some quantitative PCRs to look at canonical ISGs, or interferon-stimulated genes, to see if the interferons were actually inducing the expression of MX1, IFIT1, and viperin in the two bad cell lines in a control A549 human cells. And we see that as well. So the model's working, our recombinant interferons are functional, STAT1, STAT2 proteins do what's expected, but they're doing it faster in bad cells than in human cells, and we see canonical ISG expression. So then we asked ourselves, this is great. The interferons are working, we see some ISG expression. But what if bats express more ISGs or a different subset of ISGs than humans, right? So that's what Victoria went on to do, and I'll tell you that in a quick second. I want to point out that the coronavirus proteins have evolved to block this exact pathway I told you about. So while humans are trying to mount an antiviral response, viral proteins like all the accessory proteins, ORF7b, 3a, from both SARS and MERS can block many different components of this pathway. So Victoria took the bat cells and she infected them with MERS coronavirus, and then she pre-treated them with interferon, and she also did a prophylactic post-treatment to see if the process of interferon antagonism looks different. So I'll point out a few things. There's a lot going on, so don't look at the whole plot, just look at the boxes. So we look at phosphorylation of STAT1, a marker of an active antiviral response. And in human cells with MERS coronavirus alone, we don't see phosphorylation. But this has been shown that MERS can actually shut down phosphorylation of STAT1. However, in bat cells, we continue to see levels of phosphorylation. When you look at total STAT2 levels, in human cells, we start seeing a degradation of STAT2 as well. This has also been demonstrated. The human part's been done. But in bat cells, we don't see degradation of total STAT2. The same is STAT1. We start seeing degradation of STAT1 as we infect them with MERS coronavirus, but in bat cells, we don't. Now, some of you might be wondering about the housekeeping. I see some nodding, right? This has been shown that MERS coronavirus shuts down GAP-DH beta-actin expression. So we've now got 3 additional human cell lines to show that this is actually cell type dependent. The whole MERS coronavirus field uses Hu7 cells as a model to study, but I think the field is, this is an artifact of the cell type. We've shown in other cell types, MERS does not shut down housekeeping genes. Of course, in bat cells, there's no effect, right? But clearly, MERS coronavirus is doing things differently, the interferon responsive transcription factors in bat and human cells. And then finally, what do the interferon response genes look like? So Victoria took human lung cells. We also had a tissue type matched human primary kidney cell to go with their bat kidney cells. So RP-TEX, Paki cells are the cells from Australia, and EFK cells are the Canadian bat cells. We treated them with their respective species matched interferons, and we performed bulk RNA sequencing. And then Victoria went on to analyze the data in collaboration with Dr. Loeb and Dr. Doxey at the University of Waterloo. And what we found was very cool, in my opinion. We do see upregulation of canonical interferon-stimulated genes. So everything on the right are genes that we see in bat cells, right? But this is well known. OS1, OSL, OS2, they're all well studied in humans. What we're very interested is this 136 genes that are only upregulated in bat cells. So these include GBP1, GBP5, and we've got about a dozen genes that don't have orthologs in humans. They don't have a name. So this is what Victoria is currently working on. We've created a library of these ISGs, and we are scanning them for antiviral responses. And we found some very good hits, and that's going to hopefully wrap up the study for Victoria, where she's going to demonstrate that bats, divergent bat species, have evolved an interferon response that turns on specific subsets of ISGs that humans don't have. And she's going to demonstrate the potency of these ISGs against a range of different bat-derived coronaviruses, bat-derived pox viruses, and bat-derived herpes viruses. Good. So while she'd made all these interferons, one of the experiments, Rita came to us from the University of Barcelona in Spain, and she had this great question, like, if bat interferons are so cool, could we use bat interferons as a therapy? And she's like, Arinjay, this is a Nobel Prize for the idea. I'm not even kidding. She was very excited. She walked in and she took human interferons, put them on bat cells, took bat interferons, put them on human cells, and we saw nothing. Absolutely no protection. And she came in and she's like, Arinjay, I don't think you're getting the Nobel Prize. So we haven't made a new interferon therapy. And, but then what we decided to do, and Rita was like, well, perhaps bats and humans are too distant. Why don't we take 2 bat species and swap their interferons? So she did that. She took the Canadian bat, Heptesicus fuscus interferon, And when she puts them on the same bat cells, works beautifully. No green, complete protection. However, when she put this on the Australian bat cells, nothing. Absolutely no protection, even within bats. And then she takes the Australian interferon, the Australian bat interferon, and she puts them on the Canadian bat cells. No protection, absolutely. No protection, right? So that got us very intrigued. And Eric and I were talking about this morning. And the reason we are intrigued is if interferon is conserved across vertebrates, so invertebrates rely on an RNAi mode of innate immunity, vertebrates rely on interferons. If all interferons have to do is interact with the receptor and turn on ISGs, why is it so tightly species specific? So Rita went on to do, don't look at the whole thing, I'll bring you down to a small box. Rita went on to do a very cool positive selection analysis, and she identified two residues in interferons, the 114th residue and the 141st residue in interferon beta, that perhaps suggested it was positively selected for across different mammalian species. So we said, why don't we go test that? We had the wild type interferon. Victoria had already made it. Rita then went on to make 35 different recombinant cytokines with all sorts of combinations of mutations, and then we tested them. So what Rita found was vehicle-treated cells, you dump on different concentrations of interferon, doesn't affect VSV, replicates just fine. You take human wild type interferon, and as you increase the amount of interferons you add on these cells, you start seeing protection. Expected, we know human interferons are functional. You take interferon beta with this mutation from a histidine to a glycine, you now start seeing loss of protection, right? You see some protection, but it's not as potent. When you quantify this, it's not as potent as the wild type. Same with the second mutation. When you combine the two mutations, so this is human interferon beta that's mimicking the two residues that bats naturally have. The interferon is no longer functional. So we've narrowed it down and we've reversed the story. We've now tested the bat mutants with the human residues across different bat cell types, and we lose complete protection. So for whatever reason, human interferons and bat interferons and mammalian interferons have evolved these two residues for very specific association with something. We think it's the IFNAR 1 and 2 complex. Makes sense. The receptor evolved, so the interferon evolved. But the question is why? Why would the IFNAR 1 and 2 receptors evolve? And why would the interferons evolve? There's no need. So we've got some wild, crazy theories that we're testing, including perhaps an ancient pathogen was competing for the IFNAR 1 and 2 receptor. That forced the receptor to change, which forced the interferon to co-evolve. But I'm just hand waving. We've got no data for this. So we are exploring different sorts of stories, and I'm open to ideas. Please, if you've got ideas, throw them at us, because this is data from November 2nd, so fairly new. And we are still testing other combinations of mutations as well. So with all of this knowledge, what we eventually want to do is we want to be able to deliver them and come up with some therapeutics. It's very, our field is currently at stage A. So we are still identifying cells. We are trying to identify different ISGs. But we are working with Dr. Bowen Lee at the University of Toronto to package some of these ISGs in LNPs and deliver them into hamsters to see if they actually work. And then our goal is to also make chimeric ISGs, so human ISGs with bat sequences in them, and see if they're more potent as antivirals. So that's our 10, 15, 20-year stream, perhaps. And Kaushal is also working on some of these aspects. So I'll jump through some of the slides very quickly. I don't have a lot of time. But SARS-2, when it first emerged, while we were working on the human consequences of infection, we also set up a large collaborative study with, well, Dr. Chris Overall here at UBC as well. Just, yeah. So to show a young Dr. Overall, to show that how SARS-2 would interact with the bat innate immunity. Right, so we started this with developed primary cells, and I started this as a postdoc, and now Kaushal's taking it on. And over the last 2 years, it's wrapped the study up. We should be putting this out fairly soon. So what we looked at was innate immunity in bat versus humans using transcriptomics. Chris helped us with the proteomic component of it. It'll be the first proteomic data set from SARS-2 infected bats. But I'm only going to show you the last part of the story because, again, we don't have a lot of time. But what Kaushal observed was when you infect bat cells with SARS-CoV-2, within 24 hours of infection, the virus evolves a specific mutation. 65% of the population represents a specific mutation that changes the furin cleavage site. So we go for a PRRAR, so the last arginine changes to a proline residue in bats for SARS coronavirus 2. So it selects for a variant that has this mutation. And then we did some computational modeling studies that suggested that this would take away the ability of furin to interact with the site. So Kaushal went on to test that. And again, a lot of this work was done in close collaboration with Chris, who suggested all the experiments. So Kaushal went on to express the full-length spike, the wild-type spike, we called it. We created a mutant where we deleted the entire FCS, and then we took the bat-adapted spike as well. And when you put them in cells, we clearly start seeing, and these are done in bat cells, we clearly start seeing that wild-type spike does get processed and cleaved. However, both mutations, especially the naturally evolved mutation, takes away the ability of spike to get cleaved. We also used reverse genetics to rescue these viruses. So we had wild-type SARS-2, and we had the PRAP mutation-containing virus as well. And using Vineet's system that he developed at UTMB, we were able to show that in Vero E6 cells, there isn't a functional disadvantage for the virus that evolves in bat cells. However, in Calu3 cells, in human cells, the bat-adapted virus has a replication disadvantage over the wild-type coronavirus. And again, with Chris and others, we've now gone on to show that why this is and perhaps why the virus evolves. In fact, Francois suggested an experiment to us this morning, which will help us wrap the study up faster than we'd expected. So stay tuned, we'll have more data for you very soon. So having shown you all of this, I'll wrap up by telling you why we think bat coronaviruses are highly unpredictable. Models are great. They'll tell you which spike can interact with which receptor, but you wouldn't know for certain unless you actually do that experiment. And a classic example of that is the study where NeoCoV and PDF2180 are viruses that are very similar to MERS-CoV. MERS-CoV does not use ACE2, it uses DPP4 to enter cells. But this study very clearly showed that bat MERS-like viruses actually interact with ACE2. So every model that predicted that was wrong. And now there's a good preprint from Michael Letko's lab that shows that all morbecoviruses in bats use ACE2. So those bat-derived viruses do not use DPP4 as a receptor to enter bats, to enter humans. However, MERS coronavirus still does. There are also lots and lots of unknown coronaviruses that continue to exist in bats. SARS-CoV-2 is likely not the last one. So if you look at the most recent study that found 17 novel alpha coronaviruses, these are viruses that infect swine. So if you look at porcine epidemic diarrhea virus, some of you may not have been around in 2009, undergrads maybe, pork chops became very expensive in Canada, partly because PED-V was wiping out pig population in the US and Canada. So it directly affects food security as well, although it may not infect you individually. And if you look at this list, they also identified 7 novel beta coronaviruses. This is one study in one cave in China. And they identified 7 novel beta coronaviruses, some of them with the potential to infect and interact with human ACE2 as well. And if you look at the list of priority pathogens that the WHO put out, 7 of those 12 viruses come from bats. And disease X could be anything. So at least 7 of the 12 viruses are found in bats. So we believe it's extremely important to study these pathogens and identify what causes virus shedding from bats. We're also very interested in identifying do all bats carry zoonotic viruses? Because 1,470 species, how would you run a surveillance program for 1,470 species? But we think there are clues that we can work on. So if you look at sarbecoviruses, SARS corona-like viruses, they come from rhinolophus bats. So you could direct your surveillance efforts to rhinolophus bats only to identify pre-pandemic sarbecoviruses. And the same might be true for other coronaviruses, we just don't know yet. So I think we need to come up with a risk matrix to identify which viruses have pandemic potential. So once we're doing this, we were also looking at can species-specific restriction be a feature that bats have evolved? If not all bats carry all viruses, perhaps their immune responses are refined and fine-tuned for the viruses they carry. So we looked at a gene IgG15. It's a huge collaborative study. We've got three members of my lab who are part of this, and they developed an IgG screening assay. It's a beautiful assay that we're now going to use moving forward. And we looked at 12 different bat species. In humans, it's published and known that IgG15 does not block SARS-2 replication. It doesn't do very much. So when we tested the bats, we found actually very surprisingly that in some bats, it's pro-viral. We see more virus replication when you start supplementing the cells with IgG15, whereas in other bats, it's antiviral. So this is, again, going to support this argument or hypothesis that perhaps bats have evolved responses that are very refined and fine-tuned to the viruses that they've co-evolved with. Developing new tools, part of the reason I'm here today is to seek your help. Our field needs a lot of help. We've not done a lot of these fancy assays that you're using for rodents and humans. So if we can collaborate to take your technique into bats, we'd love to do that. And finally, I'll end with this. There's a huge gap in our knowledge between field epidemiology studies and laboratory bat studies. But our colleagues in the field have shown stressed bats can shed more Hendra, potentially leading to an outbreak. We don't really know what that model looks like. What does stress look like? How would you quantify stress? So we've started this initiative, and hopefully this gets funded by NIAID eventually, where we take data from the field and we model outbreaks using bat organoids and bat in vitro models, where if cortisol levels are high, we simulate that and see how that impacts virus replication. And finally, this is what we are interested in doing for the next 3 decades of my life, hopefully. What sort of viruses bats have? Can they infect humans? What sort of hosts can they infect? And develop diagnostics, vaccines, and drugs, because we don't want another pandemic. And with that, thank you very much. If you're at ASV, please come to us. We've got a satellite on bat virology. We'd love to talk to you. And thank you.
About 1,500 species of bats. I mean, that's the largest mammal.
Rodents and bats.
I think it's more bats than rodents.
Well, that's debatable. Rodent scientists would tell you we are wrong.
Right.
So,
but is that because, I mean, that there's not many flying beasts at night, that they've expanded so well to work at night?
I wouldn't know.
This is just owls and...
I don't know. No idea.
What are the predators of bats?
Hey, there is a paper showing monkeys, a certain kind of monkey, would eat bats off of trees. Talk about One Health and zoonotic transmission. Think about a bat infected with Ebola and a monkey just grabbing it and eating it. And non-human primates die of Ebola virus infection. So if you look that paper up, look at monkey eating bats, and there's an actual manuscript with photos of monkeys eating bats. That's just mind-boggling. But somebody did that study.
Yeah, I'm going to lean over here awkwardly. Great talk, Arinjay. I'm wondering, you mentioned a lot about the type 1 interferon response, but what about the type 2 and type 3 interferon responses? Are those prominent in bat coronavirus infections, and how do they differ from human responses?
They do have type 2 and 3 interferons, and Hannah and Christoph, 2 postdocs, were starting in the fall. One's going to chase type 2, one's chasing type 3. Because this is the first time anybody's made so many different kinds of recombinant bat cytokines. So we feel confident producing bat cytokines now. This is something we wouldn't have done 3 years ago. But now we want to make every bat cytokine and study it functionally. Yeah, for sure. No, we want to do TNFs too, because we've shown that inflammatory cytokines are not very potent, and the whole field believes bats don't have inflammation. I'm not so certain. I think there is inflammation, it's just not as pronounced. But we need to tease out those differences, and for that we need to make the cytokines. So now we will make the cytokines. So lots coming in the next 10 years. Maria.
Thanks for an awesome talk. That was really fascinating. I had a question about the persistent infections. Those are awesome and really cool to system. So for the persistent infection, do you think that there's sort of, if you were to kind of come back with another infection in these animals, do you, in terms of an innate immune response, do you think it's going to be more like a trained immunity phenotype or a tolerant phenotype? Are you going to see more ISGs because they've now been exposed for a long time with these viruses, or are they going to be more tolerant? And does that have any impact on the pathology and the pathogenesis? Yeah.
Great question, Maria. This has been done to a certain extent by colleagues who sample bats. So they have done these regression models and correlation studies where they've shown that bats that are co-infected are more likely to shed. Now, we don't know what's driving what, but white nose syndrome is a fungus that's wiping out bats in North America. So little brown bats are now endangered and threatened because they are dying out of a fungal infection. So if my former PhD supervisor showed that when you experimentally infect bats with the white nose syndrome fungus, they start upregulating transcripts from an endogenous coronavirus. So you're right, I think secondary infections can trigger virus shedding and replication. And that's one of the theories we want to test in our persistent model, reinfect the bats, keep them for 3 months, and stress them with fungal exposure or even cortisol exposure. So there's data showing that cortisol levels can fluctuate, and that affects bat virus shedding as well. There's no empirical data on it, and that's the gap we want to fill. But we don't know, but we want to test. Sure. Selena?
Just piggybacking on that question, for the persistently infected MERS bat cells, have you thought about like doing an interferon cure and then sequencing to see what the adaptations are for the persistent infections?
Yes, so Kaushal really wants to do that, but I want him to wrap up this paper first. But he does want to bring up those persistently infected cells, do two things: compare them with mock and acutely infected cells with transcriptomics and proteomics, see what the profiles look, and also to try and cure them. In fact, we are doing a bat trial this summer where we'll first infect them with MERS, and then we are going in vivo with the interferons we've made and see if we can get rid of virus replication in the bats. But that's exactly the direction we're going with persistence.
Thank you, Arinjay. Really great talk. So I'm a mouse gut person now. I think you convinced me. Maybe I should study bat now. So I have two questions. One is a very naive question. We know from mouse studies, they have coprophage. For bat studies in your persistent model, do bat have a coprophage phenotype when you see a virus in the gut?
Yeah, it's a good point. So bats poop hanging up. The poop falls straight on the ground. You're talking about coprophagy, right?
Yeah, so coprophage. They eat their own feces. Yeah.
Yeah. No, we don't see that in bats. Like behavior-wise, I've never seen a bat eat its own feces because it's a tremendous amount of effort. You have to fly down, squat on the floor, and eat it, or you have to catch it while you poop it, and on the fly you catch it, right? So I haven't seen that. That doesn't mean it doesn't happen, but we just haven't noticed it. But,
So this comes to the second question. You said the virus can persist in the gut. So have you been able to see actually the virus can be sort of hijacking the intestinal cells? They stay there forever, persistent?
Yeah. So when I give a talk at U of T, I run into a colleague who's going to help us do this assay, where you can take sections of the intestine, and then you can start mapping different cell types. We haven't been able to do that, and a huge limitation of our experiments are we do them in CL3. They're all risk group 3 pathogens, and there are restrictions on what samples we can ship and where we can ship, especially for MERS coronavirus, because it's also a security-sensitive biological agent. So we do want to do that. That's a gap that the whole field currently has. We don't know where the virus is sticking around. You take gut, and the gut is massive. Even for bats, the gut extends to, I think, about this big, if you tease it out. And you got to take every section, because you never know in which section you're going to find the viral RNA. We take the whole thing, homogenize it, and we look for virus, right? That's easy.
Yeah, I do that on daily basis. Just the whole in mouse intestine, just from different segment. Yeah.
Yeah, but we'd love to do that. If you've got a technique that you think might work, we can look into SOPs that we can probably use to inactivate the virus and ship you that intestine. But it'll be great. We do want to know for sure. Thank you. Yeah, we don't know why the virus is viremic in the blood of bats. We should test that.
So my question is, how many of the coronaviruses or how many viruses should blood people be worried about with massive viremia?
Well, every virus. Blood is really the highway for every pathogen. So without the highways, the pathogen doesn't go systemic. So I think every virus is relevant for blood. That's my naive take on blood. But without the highways, the viruses wouldn't go systemic. So.
For sure.
Thank you. Thank you.
Simple answer. Thank you very much for a really entertaining and interesting talk. I'm curious, what species will the bat initially be inoculated from?
That's a great point. So that is presuming that bats don't already have this virus persisting at the population level. If you think about herpes virus, our kids will get it from us, humans. Okay. We don't have to go to a different species. So
I'm asking you this question because I'm presuming that your inoculum in the laboratory is from vero cells?
I see what you mean. Okay. Yes, great point. They're vero-derived, and every
Vero is monkey cells.
Yes. Every coronavirus we study is essentially a human isolate. You're absolutely correct. We are trying to get other viruses from bats if BAC would actually sign off on my import permit. And then we'll have those viruses and we'll put them in bats. But great point.
Your stay tuned slide that you showed toward the end showed that Vero and Calu cells, which the latter is human, you're showing differences.
Which
you pop out a cell, or you pop out a virus from a human cell versus a virus from a monkey cell. They have a different envelope covered with different human or virus encoded proteins plus monkey encoded proteins. Yeah. Is this the basis for your Nobel Prize experiment failing?
All right, so
or
let me
not failing, but doing what you think something else.
Okay, all right. Think about this. If this is a human cell, Yep. So let's think about this as a human cell, yeah? You talk about your monkey cells, monkey-derived virus, okay? And you talk about, let's say, human-derived virus, yeah? And you're correct. The virus that we prep in the lab, stock, would have proteins on them that are human cell-derived. It goes in once, right? Everything that comes... Sure, monkey-derived. So let's even take this out. It goes in once. Whatever cells you're studying, let's say this is a bat cell. The first infection would be with the virus that's monkey-derived. You're correct. Every virus that comes out of this cell is now a bat-derived virus with the bat envelope on it, with the bat proteins on it. So I think for the first step of going in is something that would be dependent on your stock. But we're not studying that. We're studying infection across your cells and in vivo, but the virus has gone through multiple rounds of replication. So I think that would sort of take care of your question because you've got lots of progeny virions that are replicated and now doubled up in cells from that species. We don't know that. Oh, you do? Okay. I'd love to chat with you. Yeah, that'll be great. Yeah, because we don't, I have no data to back that up. So we'll talk.
I'm sorry? Yes, we have published that human-derived proteins signal cells to enhance the infection of herpes viruses.
Wow. In one cell.
Yes.
Because everything coming out will be relevant, right, for that study?
Yes.
Yeah. Wow. I'd love to read that. I'm mind blown. So that might actually have implications for zoonotic transmission as well. Because the first successful transmission event from bat to a human would include a bat-derived virus making it into a human or intermediate host, whatever. That's fascinating. Very important.
Envelope.
Yeah. Wow. Thank you for that. I'm going to go back and look at it and get my students to look at it too. It's fascinating. Good. We'll aim for the Lasker and then